Hey guys, this is our last histo lab of M1, which is kind of crazy to think about. All right, so this is microanatomy of the eye. What we're looking at is basically the three different layers of the wall of the eye, and then we'll look at the eyelid very quickly at the end. But primarily we have the outer fibrous layer, which you can see here in the cornea, and the cornea extends into the sclera. Then we have the middle vascularized and pigmented layer, which you can see here. This is the choroid and extends into different specializations such as the ciliary body, and then we have the iris right here. And then finally we have our sensory region, which is the retina, and what we're going to be dealing with is the inner retina. This is in and this is out, as we will talk about quite a bit today. And then within the inner part, we have the non-visual section of the retina, and then the visual section, which is the rest of it. Okay, and then we have our outer portion of the retina is the retinal pigmented epithelium, and again, we'll talk more about that. All right, here's just another diagram of the same thing. So we have our outer fibrous layer with the cornea, and then it becomes the sclera. Then we have our vascularized pigmented layer, the choroid, right here. And then deep to that, on the inside of the eye, we have the retina, which is our sensory layer. All right, so this slide is just saying the exact same thing. So again, cornea, sclera, our fibrous layer, middle vascularized layer, then we have our retina. Okay, so then we have different chambers of the eye. So the very large chamber right here in the back is the vitreous chamber. And you can see we have the lens here. Overall, you have the retina bounding it. And then we'll talk about the ciliary processes right here and the ligaments that they extend to the lens. But we have vitreous chamber, then we have posterior chamber, and then we have anterior chamber. The anterior chamber and posterior chamber are going to have aqueous humor, and then the posterior chamber is going to, or the vitreous chamber is going to have a vitreous humor. So we'll talk more about that as we go forward. So another diagram of just the three main layers of the wall of the eye. All right, so we're starting with the cornea, and we like to think of the cornea as the first thing that the light passes through. And so the cornea has got five main parts. On the very outside right here, we have the corneal epithelium, which you can see is in fact stratified, right? Multiple layers. It contains pain receptors, and it can regenerate. You see we have sort of a basal layer, which can proliferate and become the layers above it. So corneal epithelium, epi being top. Deep to that, we have our second layer, which is right here, and this is Bowman's membrane. It's made up of collagen. It's a barrier to trauma and bacterial infection, so it's very thin, but right here, this is Bowman's membrane. All right, deep to that, we have the stroma, and so this is avascularized and highly organized collagen. You have these cells that are embedded in this collagen, or the ones that make it, and these are the keratocytes, which are specialized fibroblasts, and they're producing type 1 collagen, which you can see here. All right, but dense, regular connective tissue. All right, so next, and again, this is the cornea, which you can is transparent. Next layer is another sort of basement membrane right here, and this is Descemet's thick basement membrane. You can see right here. And so we like to remember Bowman's versus Descemet's as B before D. So Bowman's on the apical surface, and Descemet's towards the inner part of the eye near the aqueous humor. And then you have D to that, you can see a bunch of squamous cells. This is the corneoendo, meaning inside. Epithelium, simple squamous, regulates hydration of the stroma. These cells have occluding zonules to seal off the rest of the cornea from the aqueous humor, but they're still, allowing some nutrients to go through to provide nutrients to the stroma and the layers above it. Okay, and then one last thing on the outside surface, we do have a tear film that keeps the corneal epithelium moist. All right, oh, and then the corneal endothelium cannot regenerate, and as you get older, these guys will die off, but you'll see these cells sort of spread out to maintain this barrier from the aqueous humor. Okay, so does not regenerate, does regenerate. All right, so that was the cornea, which again, see-through. Our fibrous layer continues to become the sclera. All right, so cornea is the receptor of light. The sclera is, the fibrous tunic, fibrous part that covers the posterior portion of the eye. Again, all of this region right here is sclera. It's made of dense irregular connective tissue. You can see this layer right here. It is full of fibroblasts, and around the fibroblasts, we have our type 1 collagen, and around that, we have a ground substance, which is keratin sulfate and chondroitin sulfate. Yep, so that's pretty much it. I doubt we need to know too much in that other than dense irregular connective tissue. Okay, so that's the sclera. Next, we have the portion where the cornea and the sclera meet, and so this region is called the limbus region. So again, cornea, transparent, sclera, not transparent. Where they meet, this region right here is called the limbus, and this region is fairly important because you may remember that we have aqueous humor, vitreous humor, and so here is where you're going to have draining of aqueous humor. You have production of aqueous humor right here, and then we have the muscle, the ciliary muscle that pulls on the lens, and we also have the cells that produce the aqueous humor. All right, so this region again is called the limbus. It's where the cornea and the sclera meet. Again, this is the cornea. Here we have corneal epithelium, corneal endothelium, and you can see that the cornea, the see-through part, is continuous with this region right here, which we'd call the bulbar conjunctiva, and you can see that we got loose connective tissue right here, different from the dense connective tissue below it. And so this basically buildup of loose connective tissue, as we'll see later, this is going to loop around and then become the, right here is the eyelid. Okay, so bulbar conjunctiva, this is epithelial layer on top of loose connective tissue, which is going to loop around and become the eyelid, and over here we'd have more conjunctiva. Okay, so next we have the corneal endothelium. This guy right here is continuous with trabecular meshwork. And so, as I started to allude to, we have the production of the aqueous humor from right here. We haven't met them yet, but the ciliary bodies, which are these guys. They're going to produce this aqueous humor. It's going to go throughout here and then come back in here into the trabecular meshwork, and that's going to make its way, the aqueous humor is going to make its way to this canal of Schlemm, and then the canal of Schlemm will allow drainage of the aqueous humor into veins overall. All right, so let's just quickly review. We have the limbus region, which is where the cornea and the sclera meet. Here you can see cornea. Cornea is see-through, dense connective tissue. We have the corneal epithelium, which is continuous with the bulbar conjunctiva, which is just more of this stratified, non-keratinized epithelium that continues down, loops around, becomes the eyelid, which we'll look at later. Then the corneal endothelium is continuous with the trabecular meshwork, which is the start of where we drain the aqueous humor. It goes through the trabecular meshwork, all of these little holes, filtered, and as a result of that, and ends up in the canal of Schlemm, which goes to veins overall. All right, so then here we have sclera, and notice the difference between the dense connective tissue and the loose connective tissue that underlies the bulbar conjunctiva. All right, so this was the limbus region, where we have fibrous layer meeting more of the fibrous layer. We have another area that is a fibrous layer specialization. And so again, cornea, sclera, and then when we get to the optic disc, the sclera is going to continue as this region here, which is the lamina cribrosa. And so, again, this is where the optic disc is. It's a region where the axons of the retinal ganglion cells converge and exit the eye as the optic nerve. However, they need a little bit of organization, and so basically the idea is that the retina, which we'll see a little bit later on, the choroid, which is the vascularized layer, and then the sclera. The sclera continues through this region as the lamina cribrosa, and it's a sieve-like opening which allows the ganglion to extend through it. But it's just like the rest of the sclera, type 1 collagen, dense irregular connective tissue. And what does it actually look like? Well, the lamina cribrosa, look how it has all these tiny holes. And so wouldn't that be perfect for little axons to go through? Yeah, so it's organizing the axons from, around the optic disc. And you can see this lamina cribrosa. It's all these like kind of darkish lines right here, and then all of the stuff in between is axon of the optic nerve. Okay, so not too bad. The sclera in the back at the optic disc becomes this porous substance, still type 1 collagen, that organizes the axons of the optic nerve. All right, so we've talked about the fibrous layer. Now we have the middle layer, which is the vascularized layer, and you can see it as this like dark pigmented area. It's best in this view, perhaps. And this is the choroid. It's pigmented, so we've represented here as brown. And we have a couple of specializations. We've already kind of started to talk about them. We have the ciliary processes here that's making aqueous humor, but the choroid, goes along the, makes up the wall of one of the three layers of the wall of the eyeball. But in the front, you have a specialization here, which we'd call the ciliary body, and then our second specialization, which gives us the iris, which we know the iris is full of colorful lines, right? So the vascular layer is pigmented and vascularized connective tissue. It specializes as the iris and ciliary body, and it incorporates smooth muscle cells, which we'll see are going to pull on the lens and allow for contraction of the iris to give us, opening and closing of the pupil. All right, so here's, again, just another, we're not focused on the limbus right now, but choroid, specialization, a ciliary body, and then iris. Okay, so here we start to get to the fun stuff. If we look at a nice little cross-section of the eye wall, this top part right here, we're going to talk quite a bit in detail about that. This is the neural retina. The bottom of that, this layer right here, is the retinal pigment epithelium. And then deep to that, the very darkly staining region, which you can see has blood vessels in it, this is the choroid layer, pigmented, vascularized. Deep to that, we have our fibroblasts and our dense connective tissue. Right? So the choroid is a portion of the vascular coat that lies between the sclera and the retina overall. And when we say vascularized, here we have nice vein, here we have capillaries, here we have arterioles, here we have melanocytes, and we'll talk more about that in just a second. All right, so what are the different parts of this choroid vascularized tunic? Well, we have, first off, our choroidal melanocytes, which are very darkly staining. And you think to yourself, well, in the skin, when we looked at skin, the melanocytes weren't so darkly staining, right? Well, that's because the melanocytes in the skin give off the melanin that they make, whereas these guys, they hold on to it, and so they're very dark. So these are the choroidal melanocytes. Then you have this region right here, which is actually part of the retina, but it's a good boundary of the choroid. So this right here, this little dark staining region, this is the retinal pigment epithelium cells. We'll talk more about that when we get to the retina. Here we have arterioles, and they break up to, or branch to become our capillary layer, which you can see here. These are the choroidal capillaries, choroidal capillary, arterial, and then this big guy in the center is a venule. And then we have deep to the retinal pigmented epithelium, the bottom layer of the retina, we have a very thin layer right here. It's hard to see. You can see it. It's labeled here with the arrows in orange. And this is blurry. I apologize. Let's go to this image. It's still very hard to see. There's a layer right here of connective tissue, and that layer is Bruch's membrane, Bruch's membrane. And it's located between these choroid capillaries and the retinal epithelium, retinal pigmented epithelium layer, RPE. All right, yikes. But we're doing our best, so we'll continue. All right, so choroid, now let's talk about our specialization. We are, in fact, in the limbus region, and this is our ciliary body. So in the limbus region, we have the ciliary body, we have the ciliary muscle, which is going to be like a ring and pull on the lens. Then we have our iris, so let's talk about those. Our ciliary body is, again, right here. And it's where we have the ciliary muscle. And this is the inner part of the eye. This is the outer part of the eye. Just to reference, we have bulbar conjunctiva. This will loop around to the eyelid. Here we have the canal of Schlemm. This must be the cornea. This is the sclera. We have some loose connective tissue with the bulbar conjunctiva. Then we have the lens overall. All right, but the ciliary body, this region right here, is continuation of the choroid. It's vascularized pigmented connective tissue, contains the ciliary muscle fibers, and the iris is the most anterior part of the vascular layer, which we call the uvea. All right, so more on the ciliary body. It extends and folds into these processes that we call ciliary processes, and ciliary processes are going to be what makes our aqueous humor, which we'll talk about on the next slide. But you'll notice that we have a couple of different layers. So one, look at the inside of our ciliary bodies. Doesn't it look like this region? Yeah, so the inside is more of a choroid. Then the outside is, you have a light layer right here, and then you have a dark layer. So you have two layers on the outside. This is actually continuation of the retina layer. So those two layers right there. And we'll talk more about that. That's a long lab. All right, and then you also have the zonular fibers coming out that attach to the choroid muscle, and you can see that it's attached to the lens as well. All right, so more on the aqueous chambers. This is just saying that the ciliary processes are producing the aqueous humor, ciliary processes. And you have, again, the anterior chamber, posterior chamber, and then the vitreous chamber, so not too bad. Purpose of the aqueous humor is to provide nutrients to the avascular tissues of the cornea and the lens, as well as to maintain an interocular pressure, so our nice eyeball does not collapse. All right, so more on the ciliary body. We're back. So we said that you can see that we have, continuation of the choroid into as the center part of our ciliary body. We have connective tissue. You can see we have some vessels, but more superficial to that, we have one layer right here and another layer right here, and both those layers are from the retina, so the retinal layers. And so you have the outer pigmented cells, and you have the inner non-pigmented cells, and the inner non-pigment cells are what actually produce the aqueous humor. All right, that makes sense because they're adjacent to this whole lumen area, right? The posterior chamber. So you have, why are these guys inner? Why are these outer? Well, remember that outer is this way and inner is towards the lumen. So these guys are closer to the lumen. The inner non-pigmented cells are inner because they're closer to the lumen. Outer pigmented cells are closer to the outside, so they're outer. All right, so then if the ciliary processes produce the aqueous humor, basically we'll track it. It goes like this: posterior chamber, posterior chamber, posterior chamber, anterior chamber, anterior chamber. Then it goes into the trabecular meshwork, which you can see here, and then into the canal of Schlemm for drainage. All right, and so we started to talk about the muscle in this region, but this is the ciliary muscle. It's a circumferential muscle. I feel like there's a decent chance this will be a test question. The idea is that this circulatory muscle is going to pull on the zonular fibers to get the lens to flatten out, or cause the lens to sort of become rounder. And so it's a little bit counter to what you would think. For some reason, when I think, flatten out, if this guy was going to stretch, wouldn't that be a result of this guy contracting? The answer is no. So when the ciliary muscle contracts, right, this ring, if this green is the ciliary muscle, when it contracts, it actually gets smaller, right? So it contracts inwards, and when it contracts inwards, all of these fibers become more like wiggly, right? They become less tense, and so if they become less tense, the lens rounds out, and it's actually increasing in power. And so this is what you would want for near sight. So ciliary muscle contracts for accommodating for near sight. And then the opposite, when this guy relaxes, well, if it's relaxing, it's going to go from small to larger in circumference. And as a result, the zonular fibers are going to become tighter, more tense, and it's going to cause the lens to stretch. And that's actually a decrease in power, and it's accommodation for far sight. All right. So contract for nearsightedness, relax for farsightedness. Relaxing actually causes the lens to spread out. So it's a little bit counterintuitive, I think. All right, then we have the iris, which has our colorful lines. All right, so cornea is a receptor, iris has colorful lines, our lens magnifies. All right, so let's talk about this. It's a specialization of the choroid. You do have continuation of, right, we said this is part of the retina. Outer pigmented, inner non-pigmented, that produce the aqueous humor. See how it continues, and it actually becomes 2 layers of double pigmented layer. And then that's going to continue and actually become the first muscle, the dilator pupallae muscle. But the iris is defined primarily by the fact that it doesn't have an epithelium, it has a loose connective tissue, and this loose connective tissue is going to have a lot of melanocytes, which is what's going to color the eyes. So you have melanocytes at the top, then on the very sort of inner surface, you have double layer of retinal tissue, and that's going to, some of that's going to specialize and become like muscle layer. And then here we have another muscle we'll talk about in a second. All right, so the 2 muscles we have, there's one on the very end right here, and this is the sphincter papillae. This constricts muscle. You can see it here, or it constricts the pupil, closes the hole, which we call the pupil. And then you have, so that's this region right here. Then you have the dilator pupallae, which is right here. And that is basically just a specialization of this retinal pigmented epithelium, double layer that we have. And you can see that here. So this is the outer part, smooth muscle, sphincter papillae. And this is the dilator papillae. All right, and so the nice thing here is that the sphincter papillae does, like, contracts like a sphincter, right? Makes smaller, dilator, dilates, makes bigger. All right, so pretty straightforward. All right, the lens itself, there's not too much to know here. You know, it's transparent, avascular, biconvex, and it is suspended between the edges of the ciliary bodies by the zonular fibers, right? And then it accommodates based on the contraction of the ciliary muscle in the ciliary body region, which is the limbus region. Okay, so more on the lens. On the outside, we do have an epithelium, and so this is the anterior lens epithelium. You can see that it is, squamous overall. Then as you go towards the equator, you have more of the dividing cells, and as they get closer to the equator, they elongate and fill with crystallins, which are lens-specific proteins. And as they go inwards, they lose their nuclei and organelles, but you can have, basically cells dividing and becoming the clear tissue that we see inside of the lens, okay? And then deep to the anterior lens epithelium, we have the basal lamina, which is right here and a little bit hard to see. Maybe you can see it here. All right, then we have the visual retina. So right here is the non-visual retina. Then we have the visual retina. And so visual retina is going to have many different layers. So we'll talk about that in a second, all of these layers. And then we have the outer retinal pigmented epithelium, which we saw extend here and then go over the ciliary processes and inner part of the iris. So that said, we mentioned that there's a visual retina and therefore there must be a non-visual retina. So example we were given is, imagine a ray of light coming in and it can only, based on the pupil, reach certain parts of the back of the eye. And so this part right here where the light can reach is the visual part. This part right here is the non-visual part of the retina. Okay, and so the ora serrata is where you have this transition from visual to non-visual retina, and you can see that this is the ora serrata right here. There is a change in the epithelium, right? So you have this basal layer of the retina, again, you have sort of this inner layer of the retina, and then you have this outer pigmented layer. So the outer pigmented epithelium stays the same, but the inner part is going to go from, very complex with multiple layers to just a single layer of non-pigmented cells. And, you can see that these two are what cover the ciliary processes that we talked about. Okay, so let's get into what these layers are, but think to your head, oris aurata, outer, right here, outer pigmented cells stay the same, inner non-pigmented cells go from multiple layers to one layer overall, as you go from visual retina to non-visual retina. All right, so then this is where the real magic happens, right? The visual retina, the retina is the layer of the eye that does all of the light sensing. All right, so the idea is that light is coming in, and then it hits this sort of more bottom layer, the outermost, so this is inner, this is outer, the outermost nuclear layer, and the part below it. And so this is the nucleus of the rods and the cones, and the rods are doing, white, black, cones are doing color. And so these nucleus of the rods and the cones, you have organelles, and then you have the pigments of the rods and the cones overall. All right, so nucleus, organelles, pigments. Deep to that, we have our outer layer of the retina, which is our retinal pigment epithelium. And then we had Busch's membrane, which is the outer part, inner part, inner is this way, of our choroid. Okay, then we have our rods and cones are going to extend axons up to meet with this next layer, which is our bipolar cells. And they're just going to pass along the action potential to the ganglion cells. So this is kind of like a way station, or bipolar cells. You also have in this region Mueller cells, which are supporting cells. And then, so these are axons right here. These are bipolar cells, and then you have ganglion cells, and they're going to extend their axons into this final layer right here, which is the sort of neuronal fiber layer. All right, but this is axons as well. So you have sensory region, nucleus of the rods and the cones, axons, bipolar cells and Mueller cells, axons that synapse with the ganglion cells send their axons out, which eventually becomes the optic nerve. All right, so this is just saying the exact same thing, but slightly more detail. So I think, as we'll see, the best way to go about this is just to go from inner to outer. So we have inner, we have the axons of the ganglion cells. Then we have the nucleus of the ganglion cells. Those extend they extend dendrites down to meet with the bipolar cells. So this is the inner plexiform layer, which are axons and dendrites. Then you have the bipolar cell. So this is the inner nuclear layer, inside being this way. Then you have the outer plexiform layer, again, axons and dendrites, axons coming up from the outer nuclear layer, which is our rods and cones. And then you have, deep to that, the photoreceptor layer, or organelle layer right here, and then the pigment layer right there. And then the very last layer is our retinal pigment epithelium, and it's deeply pigmented to absorb any additional light. Right? All right, so retinal pigmented epithelium, this is just, again, the very bottom layer of our retina, and it's consistent throughout the entire retina, whether you are in the visual retina or non-visual retina. So what does it actually do? It phagocytizes shed discs from the outer segments of rods and cones. It restores photosensitivity, converts retinol to retinol. It absorbs excess light to prevent light reflection. It supplies nutrients to the photoreceptors, and it also maintains a blood-retinal barrier, because we know deep to this, we have Busch's membrane of the choroid, and then we have the choroid blood vessels, right, the choroid capillaries, then we have the choroid venules, and then we have the choroid arterioles overall, okay? So retinal pigment epithelium is the only layer that is completely consistent throughout, doesn't change. All right, so here's just some more practice. What are each of these layers? I've labeled them here, but it's best to go, I think, just write them out. So we go from inner to outer. So I'm going to use we'll go with red. So this is the fibrous layer. So 5 is the nerve fiber layer, and use the same terminology. This is our ganglion. This is the inner. This is our bipolar layer, but we would call that inner nuclear layer. Then you have your outer plexiform. So again, it's very important to know in versus out. Then you have your outer nuclear layer, which we know is actually the nuclei of the rods and the cones. And then you have the inner photoreceptor layer, and this is organelles. And then you have your outer photoreceptor layer, which is pigment. And then you have your retinal pigmented epithelium at the very bottom. Then we have Busch's membrane, and then we have choroid right here, and then we can see sclera right there. Okay, so blood supply is relatively straightforward. Retinal circulation, you actually have the occasional retinal, there's a central retinal artery which goes through the optic discs and spreads across the surface of the retina. And so here we have retinal blood vessel, but this is the central retinal artery, which is branched and it goes throughout the retina. Okay, then we have choroidal circulation as well. And remember, there's like a blood retinal barrier right here, but you can still get some nutrients to cross. So you have choroidal, venule, capillary, arterial, and then here we have sclera. Okay, so not too bad. This is just a tiny bit on the rods and the cones. So rods, we said, are black and white. The primary pigment is rhodopsin, which I believe I mispronounced. And then cones is iodopsin. And they in particular are going to sense blue, green, and red. Okay. All right, so then we have specializations, and we're going to be focused on a portion that is not quite at the optic disc. This is when light comes in, you go straight to back. In this region right here, you can't see it in this cross section, but here in this diagram you can. This is the fovus centralis, and it's actually going to have a slightly raised elevation right here, which we would call the macula. All right, so this region around the fovus centralis is called the macula, raised edge that contains the fovea centralis. The fovea centralis is this depression right here. Why is this important? Because the fovea centralis is all cones, right? So where light comes in, it's supposed to hit the fovea centralis, where you have all of your color receptors, right? And so basically the idea is that as light comes in, fovea centralis right here, you can see this is distribution of cones versus rods. Cones are primarily focused in the fovea centralis where light is meant to hit, right? You move your eyeball to have light maintain that, hitting that spot. And then the number of cones drops off as you go around, and the number of rods increases as you go more peripherally. Okay, so then the fovus centralis is located again in this raised ridge we call the macula. Notice how, it appears that we have less layers. And so, just count the layers. We have the ganglion cell, inner plexiform, inner nucleus layer, outer plexiform, outer nucleus, and then we have the photoreceptor layer. So here in the fovus centralis, we have the outer plexiform, we have the outer nuclear layer, and then the photoreceptor layer only. Okay, so the idea is that cones predominate and are tightly packed. There's no rods in this region. The cells are tilted, which allows the photoreceptors to receive light that is not passed through the other cell layers of the retina. And then you have one-to-one connection with our ganglion cells, which we know their axons eventually become the optic nerve. Okay, so this is just looking at the layers again. Again, best to go from in to out. So if you want to go adjacent, you can practice your. So this would be nerve fiber layer, which we can't really see right here. But then we have ganglion cell layer, inner plexiform, inner nucleus, outer plexiform, outer nucleus, which we know is our cones in this case, and then you have your photoreceptor layer, and you have your inner photoreceptor layer, which is our organelles, and then our outer receptor layer, which is our pigment. Okay, all right, so then final thing to talk about is the eyelid. We said that we had the bulbar conjunctiva, which continued around and became the eyelid. And so it goes from bulbar conjunctiva coming off of the cornea, and then you have some sclera, and we said there was some loose connective tissue here. It circles around as bulbar, and then it becomes palebral conjunctiva, and then it becomes, and you can see we have inside of here, we have the meibomian glands, and then you have dense connective tissue in the tarsal plate. That continues to the free margin, which has our eyelids, and then continues down as the outer cutaneous zone. All right, so the eyelid has 3 zones: outer cutaneous, then it has the free margin where the eyelashes attach, then you have the inner surface, which is against the eyeball. All right, a couple things to know. We have the orbicularis oliculi muscle right here. You have this dense plate of connective tissue that is encircling all of this gland, which we call the meibomian gland. And so, yep, and then we have the meibomian glands, which produce a hydrophobic ring around the eye to prevent the loss of tear fluid. Okay, so let's get into it more. Outer cutaneous is relatively straightforward, right? You can see this is stratified, squamous, and actually non-keratinized. I think that might be a typo. So keratinized. You have hair follicles that are relatively small in this region. Here's one. And then, so epidermis, deep to the epidermis is dermis, and then subcutaneous fat below that. And so the dermis is going to have your hair follicles and some of your sweat glands. So we have eccrine sweat glands. There's one right here, non-associated with hair follicles, dump directly onto the surface. And then you have your sebaceous sweat glands, which you can see here, and that's associated with a hair follicle. All right, so then that's the outer cutaneous. It's really just skin. It's not too interesting. Then we have the free margin, which is where you have your eyelashes. And notice this prominent hair follicle, so that tells you free margin. This is stratified, squamous, keratinized, and again, eyelash is the distinguishing feature. Here we have connective tissue. Then we have, this is going to be challenging, the external root, and then the internal root. Okay, and then associated with that, we have our sebaceous glands. Remember, sebaceous glands dump into the hair follicle. Okay. All right, then we have our inner surface, which is this whole region right here. We have aka palebral conjunctiva, which is continuous to the bulbar conjunctiva. And so here we have a very thin epithelium. And you can see here, there's the epithelium. It is either simple cuboidal or stratified cuboidal. You have the tarsal plate, which is our dense, irregular connective tissue in this region. And it surrounds the meibomian glands, which we said had sebaceous glands that produce sort of a waxy barrier to keep tears from leaving the eye, right? Because you want to keep your cornea well lubricated. Okay, so as you go more and more this way, you get to like this region right here, and we have the addition of mucus-secreting goblet cells. So that would be as you get further away from the free margin. Again, free margin is right here. All right, so that's it. We have a review. So here is a review of the limbus system, and this is just saying bulbar conjunctiva continues around and becomes palebral conjunctiva. It does, that transition occurs at the fornix, which is this part right here. You have goblet cells, goblet cells, goblet cells all around here. Palebral conjunctiva, and that will continue to your free margin right here. Okay, flow of aqueous humor, we said it's produced by ciliary processes, continues, this is our posterior chamber, continues around into our anterior chamber, then it goes into our trabecular meshwork, and then into the canal of Schlemm. Once you're in the canal of Schlemm, which is so trabecular network, canal of Schlemm, which is normally a fairly obvious little space, that will continue into more collector canals, which will eventually get to the deep interscleral plexus, which contains, which would be a venous return system. All right, retinal layers, hopefully we kind of know, very thin neural fiber layer from the, those are axons of the ganglion cell, so this is a ganglion cell layer. Here we have the inner plexiform layer, inner nuclear layer, outer plexiform, outer nuclear, and then this is our photoreceptor layer. Inner photoreceptor layer is our organelles, outer receptor outer photoreceptor layer is our pigment, and then our super continuous layer of the retina is our retinal pigmented epithelium. Okay, and here's our final image. We have one more time the cornea, and we think the cornea is the transparent part of the front of our eye. It is the receptor, the first place that light comes in contact with. You have 5 different layers. You have the epithelium, which you can see is stratified, squamous, and then you have Bowman's. We said B before D. Then you have your corneal stroma with your keratinocytes, which are specialized fibroblasts, and then you have producing type 1 collagen, and then at the bottom you have your Descemet's membrane, and then you have your endothelium deep to that. All right, and that is it. Our very last histo lab of year 1 of med school. Good job, guys, and well, we'll see you in other videos. All right, bye, guys.